Hello everyone, welcome to today's webinar brought to you by Microbial Insights, and this is in collaboration with the Corrosion Institute of Southern Africa. Today's topic will be tackling microbially influenced corrosion, also known as MIC, in the oil and gas and mining fields, and this will be about leveraging molecular biological tools. We appreciate that you are taking the time to join us today, and it is my pleasure to introduce our esteemed speaker, Dora Taggart. Since joining Microbial Insights in 2001, Dora Taggart has advanced the field, including pioneering work in qPCR analysis and stable isotope probing. Dora continues to propel MI forward by collaborating with leading researchers in academia and federal agencies to keep MI at the forefront of the industry. As CEO, Dora has become a global ambassador for MMMs, regularly presenting as an invited speaker at environmental and corrosion conferences, leading hundreds of technical workshops worldwide, and co-authoring guidance documents to support appropriate use and interpretation of MMMs. Dora received her degree in biomedical engineering from Vanderbilt University. Now, before I let Dora take it away, I would like to remind you all, as always, that this webinar is being recorded and it will be available on the Microbial Insights website under the webinars section in the top menu. There are also some included informational handouts. That way you can look at those during this webinar. So if you go to your sidebar, it's under the handouts section. After Dora is done, there will be time for questions. Feel free to type your questions as the presentation goes on, and we'll try to read off as many as we can when that time comes. That way she can get you your answers. So join me in welcoming a true advocate for sustainable solutions, Dora Taggart.
Thank you so much, Marshall, for that kind introduction. Thank you, everyone, for being here today to hear this webinar. I'm excited for the conversation we can have at the end. So looking forward to your questions and getting to have this with you. So I know your time is valuable, and I hope you'll find this presentation interesting. Also, I'd like to thank the Corrosion Institute of Southern Africa for promoting this and collaborating with us on organizing this webinar. So I'll go ahead and get started. So when we think about microorganisms and how to understand the microorganisms that are present in our system, that is the core concept for Microbial Insights and the formation of the company. We've been doing this for the past 33 years around the world, and today on this webinar we have signed up a mix of people from countries all over the world to hear about this topic. So we know the importance of understanding the microorganisms in corrosion, in mining, and the goal of our company is to be innovative and to be able to do this analysis very effectively so that you can trust the data that's received and understand truly what organisms that are there. And we don't sell any products or biocides, and we're not owned by any consulting company. So we're very independent in the way that we look at the microbial analysis, and that is our focus and our specialty and our core competency that we'll be talking about today. One of the first things I want to say, I've heard this so many times in calls with clients that get reports and they're concerned, is that a lot of times they're fearful because they see that microorganisms are detected in their systems. I want to reassure everyone that microorganisms are everywhere. And so just getting an ATP reading that says that there's microbial activity, or seeing a total bacterial number that says that microbes are present, that can tell us if it's going up over time, maybe activity is increasing. But in a single instance, when you see those numbers, we don't think that there should be panic that happens at that point, because microbes can be harnessed and we can start to control the power of what's happening in these systems. Also, it's important to understand that while there are plenty of microbes that can cause problems for us and souring and corrosion in these systems, there are organisms that do good things for our systems that are necessary for these things to function. So that'll be part of what we talk about today, is both the good and the bad that comes from having microorganisms that are detected. And the main focus of my talk is going to be looking at some things that happen in the energy sector as well as for the mining industry. So we'll talk about reservoirs and reservoir souring. We'll talk about pipelines and how to do best assessment of a pipeline to understand what's going on, or a processing facility. And then we'll also talk about waste storage and tailings and biofouling and other processes, and even bioleaching that can be beneficial to us when we're talking about mining. And the key takeaways for the talk that I'm doing today, I would like for us to understand that we can start to mitigate risks if we understand the types of microorganisms that are present. And this helps us to target our treatments better, more effectively. The overall goal is to maximize the lifetime of this asset, but also to do it in a very cost beneficial way. So I think you can see that there are ways to apply a minimal amount of testing and get a lot of data that can really help you understand and manage the system and the process much better. And again, harness that power that is present in this microbes in our subsurface or in these systems, and be able to truly use that to save money in the grand scheme. So from the corporate side of things, we can have a very good cost benefit analysis from just including this simple test. So microbiology is really the foundation. Microorganisms are present everywhere. They outnumber us. So understanding these processes, what's driving MIC or souring, is really critical to future assessment. So there's no talk that can be had about microorganisms without thinking about biofilms. Biofilms are an important part of microbial communities. For those of you that know about microbes, you know that most organisms prefer to be attached, and they do this through forming these little communities that exist within these extrapolymeric substances, or it's almost like a slime that forms. And in these biofilms, it provides a protection from outside sources that may be flowing through in the pipe or present in that water system. And it also gives a way for the communities to benefit from each other. So just like when we like to live in communities and we need someone who's a baker or someone who maybe is a seamstress, that's the same thing that happens for microorganisms, is they benefit from each other. So this is really happening. And when we think about corrosion on systems, this is a surface phenomenon that's attaching as these organisms create these homes in these biofilms, and then they start to influence the communities that are in that area, so we can get pitting and accelerated the corrosion that happens when these processes occur. And that's really important for us to think about, because in the past, when we looked at corrosion, we would often think, okay, it's just sulfate reducing bacteria that's present in our system. But now, as we've grown in knowledge and the depth of understanding, we know that corrosion is not caused by one microorganism. There's a mix of processes that are occurring in these little communities. And by doing molecular analysis, we can start to understand which microbes are present and how they're interacting with each other. And this is really important if you have to treat a system. There's so many times that I look at data from these systems and a biocide has been applied, but we see a minimal effect. Well, this is because when we apply a biocide and the biofilm layer is thick and protective of these microorganisms, then that chemical application is going to have an impact, but it's not going to have as much of an impact as it could if it was disrupted, because that biofilm will give protection to those organisms, and it will only affect the biocide will typically only affect that top layer and not penetrate as well. So if we combine our treatments with a physical treatment like pigging or scrubbing of these systems, that physical process will help us disrupt that biofilm, and then it gives us maximum effectiveness for our biocide as that's applied. And before we move into these talks today, just to be sure everyone's on the same page, I want to talk just really quickly about the redox ladder. This is something that I have hanging over my desk because it's a good reminder of the effect that our geochemistry has on our microorganisms. So if you go back to like a Micro 101 class in college, we learn about this ladder and how microorganisms respond to things that are more energetically favorable. So reactions that give them more energy are going to be more robust. We're going to see those happen. So typically a lot of microbes present when there's oxygen available. And as we move down that redox ladder, we still can have a lot of microorganisms, but they're going to be different microorganisms depending on those redox conditions. So the position on the ladder is basically related to our dissolved oxygen or our ORP, and that's going to affect which microorganisms can survive and can thrive under these different conditions. So a lot of times when we have a lot of hydrocarbons or a lot of carbon and energy sources that are available to microorganisms, or there's locations that have been older and oxygen's not available, they get very reduced, and this can happen very quickly if there's a lot of microbial activity. So we'll get down to the bottom of that ladder into sulfate reduction and methanogenesis, and those, as we know, sulfate reducers and methanogens can cause corrosion in our systems. So particularly when we're talking about oil and gas, that's really an important step to understand. So let's start by talking about reservoir souring. This is a really important problem that happens globally. And when we think about what reservoir souring means, we're looking for an increase in hydrogen sulfide that's produced. And this can diminish the quality of the oil that's being produced or the gas, and it also can contribute to other problems like corrosion downstream. This has a very large impact globally. There's about $100 billion annually in damages that are estimated from a recent article that came out. And that's taking everything into account: the health risks, the corrosion and equipment failure, the diminished product quality, the capital expenditures that happen. There's so many ripple effects that come from having souring in a location. And so understanding that and being able to mitigate it and control it is very important. So let's talk quickly about how souring happens in our system. I just 2 weeks ago was in Midland, Texas, and when I was landing, I was looking at the water storage fields. And if anybody's ever been there, you know what this looks like. And those storage ponds, you can understand why there's a lot of microorganisms that are present in these ponds. And so typically we're pulling water from these storage areas. We might add some additional makeup water, and that injection well is going to be introduced into the subsurface. And in those ponds, we usually have a lot of microorganisms, and those have been sitting there for a long time. So especially in the deeper zones, you can have very reduced conditions with a lot of sulfate reducers that are present. So adding this water, we've not only added, water to be able to move through the system, but we've also introduced these microbes that will start to form hydrogen sulfide, because that's what sulfate reducers do for us. And so as this hydrogen sulfide is generated, there's also naturally occurring metals that are typically present in our system, like iron and nickel and copper. So when sulfide forms, you'll get sulfide precipitation through these metals. And so a lot of times we don't measure really well in our produced fluids that we've got a lot of sulfate reducers making this hydrogen sulfide until it's too late, until we've exceeded the capacity for the metal scavengers that are present there in that system. And so if we can start to understand this and look at some precursor markers that can be important, this can help us to get around this problem that we encounter on a regular basis, because early detection is often concealed because of these microorganisms that are present and this precipitation effect with these metals that happen. So also the other effect you have as you start to move the produced fluids up through the location, you can see biofilms start to form. Again, microorganisms like to live in attached communities, so any crevices, they will form these biofilms, and then the communities start to grow over time, and then this will slough off into our produced fluids. So we've got a lot of effects that can happen in this system, and a lot of it starts in the water storage tanks and these injections that happen in increasing these in our subsurface. So what we can do, the good thing now that we have these DNA-based tools, is we can actually dive in and start to understand who's present in these systems. And so we can use that to mitigate risk. Maybe we need to do some type of biocide prevention earlier in these systems, or develop a better strategy for how we're going to control these injection waters. But knowing which microorganisms are present, what we're seeing in these systems, and understanding the community interactions can help us to really target the treatment and be able to really do a lot better job maximizing our lifetime effect in these systems and preventing hydrogen sulfide exposure for as long as possible in these systems. So how do we test for reservoir souring? We still are learning about this phenomenon and the best markers for detecting what's happening in that subsurface condition. We certainly can look at geochemical parameters, and if we're measuring hydrogen sulfide, we know there's probably a lot more going on than we're measuring. We can also look for sulfate reducers, so we can know if they're in the injection water, if they're in the subsurface of the system. And then there's additional lines of evidence that we're finding are going to be very important. So I'm going to show you a case study of this, just a little small glimpse, and this is a study that we partnered with Raw Water to do, and we're going to be publishing and putting out more of this information, but I want to give you a glimpse of what we're seeing on the microbial side, because some of these additional lines of evidence I think are going to be really important for us, and as we can understand them better in our systems, we can start to tailor and adapt what we're doing. So fermenters are organisms that produce hydrogen, and these can be used by sulfate reducing bacteria. They also can make acids that'll be present in our system. And then nitrate reducers, if we're going to do a nitrate injection, we can look at these microorganisms to help us understand if we're really counteracting that souring. The way that we do this, just to be sure everyone's on the same page, is through a technology called quantitative PCR. So qPCR is a way that we extract the DNA. We don't grow it. We don't try to put it on a plate. When we grow things on a plate, we're growing less than 1% of microorganisms in the subsurface. We just cannot replicate conditions well enough to grow things on a plate in a laboratory. So our total petroleum heterotrophs are not a very accurate way to understand what's going on. If we try to do the BART tests in the little bug bottles, they can give us general trending, but they're not very accurate from a quantitative standpoint. So we know that quantitative PCR is the most accurate way to quantify organisms that are present. And we saw this with the pandemic, that this became a global household name in using this qPCR technology. And it became that because it is an accurate way to quantify genes or microorganisms that we're interested in. So what we'll do is take the sample. It can be a scraping from a pipe, it can be liquids from the system, it might be some of the reservoir water. We'll do an extraction of the DNA from those samples, and then we're going to go in and target populations that we're interested in. So we can look for what is the quantitation of sulfate reducing bacteria, fermenters, nitrate reducers, all types of organisms that we're interested in our system, and we'll get quantitative numbers that can help us understand what is happening in that system. So I mentioned this just briefly, but we really do not encourage anymore using serial dilutions and plate counts, just because we cannot grow things well in a laboratory, and we were having a hard time seeing correlation with what was truly happening in the subsurface. The other problem that we have, if you look at the right-hand side of my slide there, you can see what the microbial community looks like. So if we did next generation sequencing on a sample before we did a most probable number, so most probable number should be growing it, and it's going to grow all the microbes that are present in our sample. But as you can see, after doing an MPN analysis and culturing that sample, we've now selected organisms that like to grow under those conditions, and we've made our diversity change. So now we have only these 3 dominant types of microorganisms that are present in our system, and the percentages that they take up are completely different. So MPN is not a great way for us to be able to understand what is happening with the bacteria in our systems. The same thing applies for ATP. I think ATP has value in site assessment, and there's a time and place where this tool can be really effective for monitoring at the site. but it really has limited potential because, as I mentioned in the beginning of the talk, ATP is going to tell you your total bacterial numbers, and it's going to tell you the activity of microorganisms in the subsurface. So if you're doing trending and you see that increase over time, it can certainly help you know that there is a problem, but it can't help you diagnose what the problem is. Because we don't know if the organisms that are increasing are ones that are related to the processes that we're concerned about. Are these bacteria that could be harmful for our system, or are these supporting organisms? So that's where the benefit of qPCR to be able to get that depth of information really comes into play. So let me show you that quick case study that I was telling you about. And this is a way I think we can really use molecular tools to help us understand our souring risk. So in this partnership, there were some bioreactors that were set up to mimic the reservoirs. One was injected with a synthetic brine and sulfate. One was untreated, and there was a group that was injected with synthetic brine. One was untreated, and then another group was treated with nitrate. And what we did in this study was to extract the DNA at different points along the process of this produced water so that we could look at qPCR, and we could also do some sequencing so that we can understand what microbes are doing and how they're changing in these bioreactors over the course of the study. So in this first slide, you can see the total sulfide formation that is happening that was plotted over the weeks of the study from July until October. The blue line is no treatment, so you can see we have an increase in total sulfide production over time. The bottom line is the nitrate treated bioreactor, and you can see that it remains pretty flat over the period. So let's look at the microorganisms. This is where I get excited about seeing this data. What we can see is if you look at the middle group, let's focus on that total bacteria, EBAC. So eubacteria, or total bacteria, we see was pretty consistent. But if you look at those other organisms and genes that we're looking at in the system, you can see that there's some pretty dramatic differences. The blue ones are the no treatment control bioreactor, and then the reddish colors are the 1, 2, and 3 month nitrate treatment ones. And you can see from those bars, we'll talk about sulfate reducers in just a moment, but look at the fermenters. You can see in the no treatment, there's very low detection of fermenting microorganisms that are present. And but in the nitrate treated system, we see a lot more abundance of these key biomarkers that are present. Also, denitrifiers we would certainly expect when we're adding a nitrate treatment, and so this is confirmation that we are getting the organisms we would expect in the system. So let's talk more about the APS, and this is a good reminder of why we want to trend data over time. So from the first sample, the nitrate treated, it really seems like the problem but over time the APS increases dramatically. So you can see in the beginning, this blue bar is the sulfate reducers with no treatment. The red bars are the sulfate reducers with nitrate. And you can see the trend lines I showed you in that first graph. So at our first collection point, you can see we have a lot of sulfate reducing bacteria that are in both locations that are being detected. And then over time, we see that the sulfate reducers that don't have treatment tend to increase over time, whereas the ones in the nitrate amended system stay pretty consistent in that reactor. So the other thing that we did in this was something called next generation sequencing that I'll talk about briefly at the end, but there were 2 key organisms that we watched over the period of this treatment. So the yellow bars are disulfotigium that was present in the system, and the blue bars are a Marinobacter that was detected. So the disulfotigium is a sulfate reducer that was really dominant in that untreated bioreactor. You can see that over the time. The Marinobacter is a denitrifier that was present in that nitrate treated system. So importantly, what I want to point out here is this disulfotigium is increasing over time, even in the treated bioreactor. And so while it was being more controlled than the untreated bioreactor, it's really important to notate this for a producer, because if we were to stop the nitrate treatment or if something was missed, what we're going to see is that disulfotignum is able to switch from using nitrate to using sulfate, and you could get a large amount of sulfide generation that could happen very quickly. So knowing this can help you be predictive of what could happen in your system if things are not controlled. So again, there's so much that happened in this study that will be coming out, and I would encourage you to kind of watch for this publication or reach out if you would like more information with this study that was done. But both qPCR and NGS were used to provide valuable data that blended well with the chemistry of what was happening in these reactors. We learned that trending the data over time was really critical, so we didn't misinterpret what was happening in the system. We also learned that nitrate was much more effective than we even expected it to be, because it really helped to control some of these organisms like the disulfotignum that could have been more dominant in that system if nitrate was not present. There are a lot of organisms, not just this one, that can utilize both nitrate and sulfate and kind of do a switch if they need to. So it is really important to understand our microbial community under both conditions so that we can do better in the prediction for our risk assessment modeling. So hopefully you guys will have some questions for me on this as we move into the question session of the study. But I also want to take a moment to talk about microbiologically influenced corrosion and what we have learned about that over the last couple of decades. So when we think about MIC and we're thinking about deterioration of metals or materials that are present in our system by microbes, we know from studies and research that about 40% of all internal corrosion comes from microorganisms, and that calculates to at least $2.5 trillion globally in loss. So a very important problem for us to address and understand, and it is a very complicated situation to try to assess and to understand and evaluate. Because when you look at the mechanisms for MIC, there are quite a few microorganisms that can use MIC or can create MIC in our systems because of the processes that are occurring. So you can have these biofilms that form along the pipeline, just like we saw in the reservoir souring. These microorganisms in the community are interacting with each other, so they're taking the organic substrates that fall through, flow through the system, they're generating these organic acids, which can have surface interactions with the metals and cause problems on its own, but it can also be used to feed other microorganisms that can cause accelerated amounts of corrosion. And we know to date quite a few of the biomarkers that are important in our system so that we can start to look for this effect and we can be more predictive in what's happening. We know some genes that are important for direct interaction with the metal surfaces. We know sulfate reducers and methanogens can cause increased rates of corrosions in our system, and we're learning more all the time about these microorganisms that are present. So the real key that we found is it really takes monitoring populations over time. One snapshot is good because we can see kind of what the system looks like, but the power comes in trending it over time so we can see if something, hopefully the goal is to see if something's going to be a problem before it happens, but we can also start to understand what our mitigation needs to be. This way then we can start to select the right biocide, the right strategy. How often do I need to inject? Which locations need the most injection? So that can come from the abundance of microorganisms that are being detected in certain areas. The biocide evaluation is really important because a lot of times biocides are only tested on sulfate reducing bacteria. Methanogens have different membranes. So understanding the types of microorganisms that are present in our system can help us to be better in our selection so we get maximum impact in our systems. And again, this is going to help us maximize the lifetime of that asset and prevent failures that can occur. So how do we test for MIC? So we're going to go back and talk about qPCR again, but this time, rather than doing qPCR on a single target or a single group of organisms, a single functional gene, we're going to be talking about looking at a lot of mechanisms, because there are a lot of community interactions that happen in MIC that we need to understand. All those different processes to do a good job of interpreting what's going to happen in our system. So because if we tried to do little individual tubes and run each of those reactions, it would take an enormous amount of time and an enormous amount of sample. So you'd need a gallon of water and months to be able to get through all of that analysis. But with a tool called Quantarray, this is how we can analyze and look at multiple genes and pathways that we're interested in all at the same time. So the way that it works is we extract the DNA directly from the sample again, but rather than doing a qPCR amplification in a little tube, we're going to put it on this platform. You can see this slide here has little squares. Each of these squares is a sample, and each of those samples has a subarray, and those subarrays have 56 through holes. So we can do 56 individual qPCR reactions all at one time on a single sample. So we get quantitative abundance of all these different genes that we're interested in, and that we don't have to worry about them interacting with each other because they're happening in a nanoliter volume in these little through holes all independently. So when we get the data, it looks like this. It can feel a little overwhelming when you see all of this, but it's pretty simple once we start diving in to interpret this data. Because what we'll do is we can look at the total bacteria, total archaea that's present at each sample in our system. We can then start to look at groups of microorganisms that we know are important for corrosion. So are we seeing sulfate reducers that are present? Do we have methanogenic organisms present? Do we have this MIC hydrogenase or TATC translocase gene that is present in our sample, because we know those can directly be implicated for corrosion? Then we can start to look at other processes. What if we have a sample in our system, as you can see here, that looks odd? The iron oxidizing bacteria are only present in the sample 4 location. We don't see it in the other 3 samples. So that we can start to say, what is different about sample 4 compared to these other locations? So in this case, we did not, or the system operator did not realize that there was any oxygen that was present in this system that could be accelerating corrosion, but there was some oxygen ingress that was happening around this sample 4 location. So this was a good indicator that they needed to do a little more evaluation of this particular location to understand what's going on, and that explained some of the increased pitting that they were seeing in this area. So while it's a lot of data to look at, it's very digestible, and you can plot it across a system so you can see how the microorganisms are changing or growing or evolving as you move through that system. But I think it's a really powerful tool for us to use in assessment. The other thing that I would highly recommend doing is we have a database of over 250,000 different microorganisms that have been collected from samples all over the world. So what we send with each sample is this heat map that you see here, and it can tell us if a population of a particular organism or gene is high, medium, or low compared to these other 250,000 samples that have been collected. So when you look at those iron oxidizing bacteria that were 10 to the 4, is that a high number or not? This heat map can show you that that is in fact a higher concentration than we typically see in other samples. So it can give you power and context to the data to be able to interpret that more effectively. So again, this database has a lot of information. It's more than 30 years of qPCR data. We have samples collected from all 7 continents, more than 60 countries around the world, all 50 states in the US. So it's a lot of information that can help you add context to what's going on. So you can say, you know, I have ammonia oxidizers that are present in this system. How does that compare to the other samples that have oxidative processes? And so you can start to kind of zone in and look more in depth at what these percentiles mean to your particular system. The other tool that has a lot of value to us, particularly when we're talking about corrosion, is sequencing technologies. And I think most of you have probably heard of this as it's become more prominent in use. Typically, when we talk about sequencing, we're talking about 16S or next generation sequencing, where we look at the name of the organism. So what is the taxonomic name? This is Desulphiromonas. This is, a Pseudomonas that's present in our system. So it tells us who is there. We can also do a technology called metagenomics, where we look at all the genes present in the sample, not just focused on the 16S portion of the gene sequence. And this gives us not only the name of the microorganisms, but the functions of what they can do. So these two tools can be very valuable, and it produces a massive amount of data that you do have to go through and try to interpret. But when we do that, there's a lot of power in what we learn and understand, just like in that reservoir souring study, where you saw that we could detect the disulfatignum and be able to track that over time. So the way the results look is you'll get the sample, how many reads were detected in that sample. So you can see sample 2 had almost 2 million reads that was detected. And then you'll get a genus level information about, when we're talking about next generation sequencing, you'll get genus level information that'll show you the names of the microorganisms and the percentage they were of the community, and then descriptions of what they can do. So like this first organism is a thermophilic organism that is anaerobic, so that tells us information about what the organism can do. It also, we can show like it's been detected commonly in corroded pipelines and hypothermal sediments. So it gives you some information about what's going on in those systems. And then the real power to sequencing, because it's impossible to look at, unless you guys are much better than I am, to look at 2 million names of microorganisms and figure out how those relate across abundance of samples is pretty impossible from the human perspective. But using statistical analysis, we can start to group them together. So we can do things like principal coordinate analysis, so we can see where are the differences between samples, which groups of samples are most similar in our pipelines or in our assets. Or we can do things like hierarchical clustering, where we can get that grouping and kind of zone in on where there's differences in the system, so then we can understand which genera are different. We can do the same type of thing for whole genome sequencing as well for our metagenomics, and do this on a functional scale point. So rather than the names, we're looking at more the functions and how those functions cluster between different groups of microorganisms. These two tools are really complementary. It's not typically an either or in the use of these tools, because next generation sequencing gives us a very high level view of what's happening in our systems and our microorganisms, so we can start to do exploration. Which microorganisms are important? How are they shifting over time? How are they shifting across a system? And we can start to learn a lot of information. It is data rich, so it can be very difficult to interpret if you're not a microbiology expert. So this is something that if you're new to the tool, you might need a little help and guidance in your first interpretation of it, but it is a great method, and we're using it a lot more in research studies to understand in corrosion, especially the metagenomic approach, which functions are we seeing in higher corrosion systems. Then we can start to look at these microorganisms, these processes, and start to design assays that we can use that can help us be more predictive. So NGS is kind of the start to help us identify what's there and what's present in the system, and then tools like qPCR with the census or the quantarray can help us quantitatively evaluate how these microorganisms are performing across our systems. So if we can start to establish some trends, we understand if there's troublesome populations that we might want to monitor more closely over time. We see a lot of differences across systems, and this can be really predictive where, as I mentioned in that one slide, we can see that there's, oxidative conditions that are happening in an area that maybe we didn't recognize that before, or the site manager didn't realize that was happening. So it can give us a lot of value in our interpretation. It's also really valuable. Again, we don't sell any biocides, but we do a lot of work with the vendors who do, so we can look before and after treatment. How do the microorganisms respond? If we change our operational conditions, what are the microorganisms doing? And then we can start to be more predictive in what could happen in the future. So let me give you a quick case study of this. This is a biocide screening study. It was a closed water tank that was produced water, and it was being treated monthly, and the water was sitting in the tank for about 4 days as a batch. So they put some corrosion coupons that were in this produced water tank, and we looked at those over time. So we had a baseline and then some post-treatment samples. And so we looked at the pitting on the coupons and the internal corrosion damage, as well as geochemistry and the microbiology of the system. And so due to time today, I'm not going to get too deep into it, but I do want to show you an excerpt of the data from the QuantArray MIC that was produced. So you can look here, the blue is our time point 0, the orange bars are time point 168 days. You can see over the time when the biocide is applied, we see not a whole lot of difference in our total bacteria. Our total archaea look pretty similar. Sulfate reducers look very similar. Methanogens look very similar. And we actually see some fermenting bacteria. These AGN through FER are organisms that can produce acids. And so, we see, some are becoming more prevalent as the biocides being applied and the community is shifting. And when we looked at the system, we did have acetic acid that was being detected at around 74 milligrams per liter. So this does seem to be valid, good additional information. Sulfur oxidizing bacteria, you can see, are pretty consistent. Iron reducers that are grouped here on the right-hand side are pretty consistent. And again, metal oxidizers, pretty consistent. So even 168 days post-treatment, we really don't see that the biocide was very effective. So what we think is that likely this biocide just didn't penetrate the biofilm, and so we didn't really see an increase or decrease in the change of MIC-related organisms, and in fact we saw some organisms be stimulated and more acid production happening post the application. So this is a great tool that you can use alongside the treatment strategy to really understand if you're getting that anticipated effect. There were a lot of populations of bacteria that were present, so there definitely was a substantial MIC threat that was present in that system, and that was why we were probably seeing that pitting and everything that was occurring in the locations, and a lot of organisms that needed to be monitored and observed. So as I wrap up today, before I take your questions, I want to talk about one other special consideration that I think is really important, and we're seeing more and more importance globally for mining and microorganisms. So where do microorganisms fit in the mining industry? So microorganisms are really powerful in changing the state of a metal. So they can either mobilize metals because you change the pH and the balance in the system. As microbes become more active, things can become more acidic or more basic depending on the processes that are occurring. And so either metals get mobilized or immobilized. And so there's pros and cons to both things. And if we really start to investigate and understand this, and we can control parts of our process. Maybe there's areas that we want to mobilize the metals so that we can recover more of those metals. Or maybe there's other areas that we don't want it to leach out of a certain area. So we want to make sure that we are keeping a certain condition so that we immobilize these metals. Microbes are really, really important in that. And so we'll talk a little bit about that this morning. One of the big problems that we see in a lot of systems, but most especially in mining, is biofouling that can occur. These mining environments have so many different metals and other elements that are present, which is why they're great for mining, but this can be harmful to microbes. And what it does is it causes a selective pressure for some organisms to be able to survive and thrive and grow, and other organisms to be suppressed. So we can get, prime environments for certain organisms like metal reducers and oxidizers, sulfate reducers and oxidizer, acid producers that can be present, and they will grow and form really big biofilms in these systems. So a lot of the times we can have these processed water or wastewater treatment areas really become clogged with biofouling from the microorganisms that are present. So again, if we want to treat this problem and prevent it, we need to understand which types of microorganisms are present so that the right biocide is selected for the treatment. The other point that I think is really becoming more and more important globally is this rare earth element recovery. And there are some great companies that we work with that are doing these recovery processes, creating cultures and microorganisms that can help enhance recovery. But of the 15 known important rare earth elements that we want to look at, there's a lot of research that's being done to understand what their natural roles are in the subsurface, how microbes interact with these elements, and then how we can better recover these in an environmentally friendly way or accelerate the recovery of these elements so that we can maximize the impact that we're having. So there are natural processes that can be done for this, like bioleaching, where we can use these microorganisms, or maybe you can add some of these cultures that are being developed that will colonize the surface of the mineral. And through metabolic activity, they'll release organic acids that help to leach out the metals or dissolve minerals. And so then we can start to recover those a little better. And we know the names of quite a few of the bacteria and fungi that can do these processes. So then we can start to track them and understand them in their system. There are also some organisms that absorb the metals, so they can be important for removing toxic metals from solutions because they act as binding sites and they bring it into their cell membranes. And so again, we know a lot of these organisms, so we can start to look for and monitor these different types of bacteria that can be present in our system. And then bioaccumulation, you can start to get some organisms that will actually take it into their cells and it can bioaccumulate. So you have a pretty decent amount of these elements that have actively passed through the cell membrane or the cell wall and are present in the cell. So a lot of information that researchers are doing right now, and through that gives us the ability to develop qPCR tests that can help us track and better understand these organisms. So right now when we do the testing, so some important organisms I've listed here like Acidothiobacillus, Burkholderia, Aspergillus, some fungi that are important. Organisms that produce biosurfactants are important for us in these processes. So we're learning more and more about them. And as we learn more about them, right now we can use things like sequencing to see what the abundance of these organisms are. But we can also, as we learn more, like on biosurfactants, we have a good understanding of the genes for biosurfactant production. So we can go in and start to use qPCR assays to test and quantify the abundance of these biosurfactant producers that are present in our system. But as a site manager or an operator in these locations, we can use these tools to help us control these processes and design our systems very effectively, or bio-augment or biostimulate with different things to create the conditions in these areas that we want to maximize our recovery. So as I close out today, these tools I think are very, very valuable. Typically, they've been very lab-based analysis where you collect a sample from a system, you ship it to the lab, and we analyze those samples for you and send you the results. But as things are evolving, we've learned that traveling with samples can be really difficult. There's a short hold time when we collect a sample. Until you extract the DNA, those organisms, those communities are still continuing to interact and change. So they're until the DNA is extracted, they're not stable. You're changing your conditions. So shipping the samples overnight to the lab, keeping it on ice so we minimize microbial activity that's happening in those samples is really important. And there are times that that's just not possible. So for instance, we've worked with NASA over the last 30 years, and if you're bringing samples back from the moon, it can take up to a year, months to a year to be able to get those samples to the lab for analysis. So what do? Those microbial communities are going to change. How do we understand what those microorganisms are doing? If you're on that oil and gas platform and there's no way to get that off and get it to a lab within 24 hours, we needed a solution. So It's taken 20 years for us to come up with this, and it's something that I've worked on my entire career, is being able to develop an extraction kit that we could use in the field. So whether it's the International Space Station or future work that we want to do for colonization of Mars, or it's right here on Earth on one of these oil and gas platforms, we need to be able to extract the DNA in a very simple, controlled way in the field so that we can have that stabilized DNA molecule that we can then ship to the lab later for analysis. So we do finally have the potential to be able to do that in the field, and we've been using this at locations all over the world for the past year. And it's been very successful. We get really good DNA recovery. Can be water samples, solid samples, swab samples. It takes about 45 minutes to extract 6 samples in the field. It's something that's written with no need for electrical equipment. It's very simple instructions, no matter what language that you speak. Even if English is not the primary language, we've had people in other countries that have been able to use these protocols very effectively. If you have minimal training, you're not a scientist, you're a field sampler, we've tried to write these protocols in a way that makes it easy for people in the field. And so no matter where they are, if it's an astronaut that's trying to do this quickly, or if it's someone on an oil and gas platform, these kits are very easy to use and get your DNA extracts. So this gives us the ability to evaluate equipment failures much more quickly. So if we saw a failure in the field, a field crew could take a quick sample of that, and rather than trying to get it shipped out to the lab, they could extract that sample and then just keep that DNA on ice, and that's stable for weeks to months. And then we can use that for further investigation later as things are moving forward. So the other concern that we have is, what if we need to know an answer very quickly? We don't want to wait to ship it to the lab, so we can have additional information and trying to that we need maybe in a 24-hour period, and we can't get it with traditional shipping methods and waiting for the lab analysis. So that brought about also, now that we can extract DNA in the field, we have a field capable qPCR system that is available for use. It comes with a cell phone, like you can see here in the picture. It's about the size of a Bluetooth speaker. It is a great tool where you can do 9 samples, 3 different qPCR targets in about an hour. So you can get rapid results that are uploaded to the cloud, give you the data back very quickly, so you can start to prioritize your sampling, sample at different locations, get an emergency response information. I used this in the train derailment that happened in East Palestine, Ohio a few months ago. So there's a lot of opportunities for how to implement this device, and it gives you really quick access to some DNA data. Then you can take that remaining DNA that's left over and send it to the lab for additional analysis, depending on what you see in those initial screening samples. So the way that it would work is to take the kit with you. When you see a sample that needs to be collected, use the bioextract to be able to isolate the DNA. You take a couple of microliters of that DNA, put it into little tubes that have the assays that we've prepared for you specific to the questions that you're trying to answer. You put it into the instrument, run the analysis, get your results back, decide which samples you want to do further analysis on, and then you can ship those to the lab. Again, they're more stable, ready for you, but you can have some initial actionable data that you can use, but you can also preserve samples for future testing. So in my career, I've been lucky enough to see us move all the way from these plate counts, which were not so fun to do for any of you who have been in a microbiology class, to PCR, to quantitative PCR, to doing it on a platform like Quantarray, and now to the field. So I feel really blessed to have seen this evolution, and it's exciting. Not only are we seeing the future as to where we can do more things in the field, get more depth of knowledge, but we're also learning about more biomarkers that are present. We're part of a joint industry project with several of the major oil and gas companies, and our goal is to identify some new biomarkers. We're doing some proteomic analysis of different systems around the world and trying to find common denominators in these degradation processes that are happening, so that we can start to understand corrosion and other concerns that will happen in these systems as well. So very exciting project. So hopefully we'll have some results for you coming up shortly in the next year or so. But really, it's our goal at Microbial Insights. And again, I'm just really thankful to Carissa for collaborating with us on this talk, but we really want to turn problems into possibilities. I think as an industry, we've learned so much about molecular biology and microorganisms and what we can do. So we need to work together and take an interdisciplinary approach to be able to solve problems for the future. So I would encourage you, if you have questions, reach out to us, reach out to your team, even if you're not going to do any molecular analysis right now, or you have an odd system or an odd question. We're always happy to talk to people and to look at data and try to help you understand what is happening in a system. And just as I wrap up today, we're seeing this more globally. Last week I was in Brazil, next week I'll be in South Africa. This really is a global concern, and we're seeing people use molecular techniques to help us address these problems. So if you do have things that you're interested in with Microbial Insights, as you can tell from my accent, I'm based in Knoxville, Tennessee. So most of the samples are in the US, but we also have a lab in Canada, one that we just opened last week in Brazil. We have 3 in Europe, South Africa, China, Australia. So no matter where the project is in the world, there's lab locations that are close by to try to make it easier for the groups to be able to do this analysis. And again, we're just seeing global uptake of using this type of technology, so it's very exciting. So I know I've gone pretty long because I had so much information that I wanted to share with you today, but I would love to have questions from you guys if there's questions that you have, and would love to take your questions now.